Okay, the slide is visible. My dear friends, the slide is
Yes,
visible.
sir.
Okay, shall we start this class, my dear friends? Anjana madam garu.
Yes, sir. Yes, sir.
Okay, start చేసుకుందాం.
Okay.
రికార్డ్ స్టార్ట్ చేయండి.
చేశాను సార్.
Okay. Okay, very good evening, my dear friends. Welcome to the session. So, yesterday class we have discussed about the nitrogen metabolism. In that nitrogen metabolism, we are clearly discussed about nitrogen cycle. So, how many processes, how many ways of nitrogen fixation? Maybe the atmospheric nitrogen fixation. Also, the second one is the industrial purpose nitrogen fixation, and the third one is most important, that is biological nitrogen fixation. So, in these three ways, three types of the nitrogen fixation. So, one of the important area and most useful topic today we will going to discuss today's class, that is the biological nitrogen fixation. So fixation of the nitrogen through the physical mode or chemical mode or the microorganisms which they are involved in this process. Okay, so in this process, uh, microorganisms, mainly the microorganisms which they are belongs to prokaryotes, especially bacteria and cyanobacteria like members, even though some of the eukaryotic members like fungal members also involved. Fungal members also involved in this biological nitrogen fixation. So in this biological nitrogen fixation, so nitrogen, the molecular nitrogen which is present in the gaseous state in atmosphere, largest amount of the nitrogen gas, that is 78%, which they are present in atmosphere. Am I right? Are you agree or not? How much percentage?
Yes, sir.
How much percentage present in atmosphere nitrogen? 78. Very good. 78% of the nitrogen present in atmosphere, it is a gaseous substance. It is a molecular state. It is in molecular state. So this dinitrogen gaseous substance is unable to utilize by all higher plants. So the unusable forms is converted into usable forms by the help of these microorganisms. The microorganisms may be the bacteria, the microorganisms may be the cyanobacteria, the microorganisms may be the fungal members. So these living organisms which they are involved in conversion of the atmospheric nitrogen into usable forms like ammonia, whatever it may be, nitrates and nitrates. These are the forms, usable forms. So these usable forms is converted with the help of living organisms. So the biological being or biological involvement, the dinitrogen, which is a gaseous substance which is present in atmosphere, that is converted into usable forms. So this nitrogen fixation we can call as biological nitrogen fixation. And my dear friends, remember here the availability of the nitrogen, which is a limiting factor of the all plant growth. Why? Because the dinitrogen is not directly used. The plants they cannot directly use. Just they are used in the form of other forms of nitrogen like ammonia, nitrates, and nitrates. Even though ammonium ion also. So the ability of the fixation of the living organisms, they are mainly prokaryotes. Okay, so they are bacteria. So the bacteria may be the symbiotic bacteria, Maybe they are not associated with any other living organism. They are free forms. They are lived outside the environment. So the bacteria which they are free living bacteria, or the bacteria may be the symbiotic bacteria. They are associated with higher plants. They are associated with higher plants. So these bacteria can fix it from atmosphere. Fixed from atmosphere. So the first attempt to increase forest growth through nitrogen fixation in Lithuania in 1894. So especially lupites in Scots and pines. Okay. So in this biological nitrogen fixation, some amount of the energy is also necessary, even though some electrons and protons are. The slide is visible. Biological nitrogen fixation. The chemical equation is visible, my dear friends. The chemical equation is visible. Is the chemical equation?
Yes, sir.
Somebody are waiting, sir, to admit.
Huh?
Somebody are waiting to admit, sir.
అమ్మా నాకు వినపడలేదు మీరు చెప్పింది. వెయిటింగ్
అడ్మిట్ చేయరా సార్? మా ఫ్రెండ్స్ వెయిట్ చేస్తున్నారు.
మరి ఎవరు చేయాలి? మేడం ఉన్నారు కదా, మేడం చెప్పాలి.
ఓకే జాయిన్డ్ మౌనిక.
ఓకే, ఇప్పుడు ఈ సెకండ్ స్లైడ్ కనబడుతుందమ్మా?
ఎస్ సార్.
ఓకే, ఈ కెమికల్ ఈక్వేషన్ ఉంది ఇక్కడ చూడండి. సో ఇన్ బయాలజికల్ నైట్రోజన్ ఫిక్సేషన్, the all microbes like bacteria, cyanobacteria and other fungal members also involved in this biological nitrogen fixation. A special type of mechanism is also involved in this process. The mechanism is nitrogenase enzyme. So nitrogenase is an enzyme which is involved in the biological nitrogen fixation. In this biological nitrogen fixation, nitrogenase is contained a special type of what we can say that nutrients or special type of minerals, that is molybdenum and ferrous. So molybdenum, ferrous units are present in nitrogenase enzyme. Along with this nitrogenase enzyme are the genes which they are control the nitrogenase activity. The genes we can call as nitrogen fixation genes or NIF genes. What we can say that? NIF genes. Okay, here in this slide we are observed here. So nitrogen. This is the molecular nitrogen or dinitrogen, and 8 flavonoxyl. 8 flavonoxyls are necessary. At the same time, 8 protons are necessary. Along with this, how many ATPs are necessary for this biological nitrogen fixation? 16 ATPs. How many ATPs are necessary? 16 ATPs are necessary. 8 protons and 16 ATPs are necessary. Very, very important. Even though competitive examination also this how many ATPs are important involved in this process. At the same time, how many protons are involved in this biological nitrogen fixation? Come on, tell me how many protons are involved? How many protons and how many ATPs are involved? Come on.
16 ATPs and 8 protons.
Online class lo uన్నా. డెఫినెట్లీ అన్న, అరే మీరు చెప్పిన తర్వాత వాళ్ళ తిరుగులేదు. అన్న ఇట్లేదు అట్లా ఫాలో అయిపోదాం. ఓకే, షూర్ రైట్ అన్న, థ్యాంక్ యూ. రైట్. So in this process, my dear friends, 8 protons are necessary. At the same time, 16 ATPs are mandatory for fixation of the nitrogen from atmosphere in the biological nitrogen fixation. So 18 ATPs, very, very important. At the same time, this nitrogenase enzyme is also, along with this nitrogenase enzyme, magnesium ion is also involved or utilization in this process. So then, dinitrogen, 8 flavonoxyl and 8 protons and 16 ATPs and 18 H2O molecules. H2O molecules means 18 water molecules are involved in this process. So this nitrogenase enzyme, it is converted into, see here, so dinitrogen or molecular nitrogen is converted into two ammonium ions. Two ammonium ions. Okay, two hydroxyl ions, 8 fluoroxyl. Let's see here 16 ADP. So 16 ATPs are involved, utilized, and 16 ADPs are here, and phosphate. So phosphoric acid or phosphate 16 H2PO4 is released. See, ultimately, the what is the final product? End product here, two ammonium ions. So one dinitrogen molecule is converted into how many ammonium ions? Two ammonium ions is released, or two ammonia atoms are released. So this mechanism is we are observed in microbes like bacteria, cyanobacteria, even the fungal members also. This is internal mechanism. This chemical equation completely explains how dinitrogen of atmospheric dinitrogen is converted into ammonia or ammonium ions. Okay, see here. So this rare and extremely energy consuming conversion because of stability of triple bonded end. See here, my dear friends. Here, I'm unable to write here. So in this process, so in between two nitrogen atoms, triple hydrogen bonds are strictly pointed. So these triple bonds is diluted or converted or break down by this nitrogenase enzyme. So in this process, 16 ATPs are required. How many ATPs are required? 16 ATPs are required for this energy consuming process. Are you agree or not? So this dinitrogen or molecular state of nitrogen is going to break down between two nitrogen atoms. Triple hydrogen bonds is break down by the utilization of 16 ATPs energy. Am I right or not? So in this process, this produces nitrogen which can be directly assimilated into nitrogen containing biomolecules, that is ammonia. Okay, here the third slide, I hope you clearly visible. ఈ స్లైడ్ కనబడుతుందా? ఈ మూడో స్లైడ్ కనబడుతుందా?
Yes, sir.
Okay, right. So in this image, we are explain. So what you observed, come on, what you observed in this slide, clearly explained. Very with our naked eye, we are easily understood this image. Come on.
పెళ్ళికి ఏమైనా అవసరం ఉంటే తీసుకోవాల్సి వస్తుంది కదా, మళ్ళీ పెళ్ళప్పుడు కూడా అవసరం ఉంటే తీసుకొస్తే.
Okay, see here in this slide, see here dinitrogen molecule is converted into ammonia. So this is the nitrogen fixation process. In this process, whether the bacteria they are associated with the symbiotic association associated with the legume plants, or some of the azospirillum bacteria which they are associated with non-legume plants, and also some of the bacteria they are lived in free living manner. They are lived in the soil. Am I right? So the best example of symbiosis, the bacteria which they are associated with the root nodules. See here, so Rhizobium leguminosarum, and see here the second image. This is azospirillum, and this is rhodospirillum. Am I right? See here, so energy source of organic carbon. Energy source of organic carbon, it is absorbed from host plant here, and here root exudates from the host plant. But here heterotrophs, plant residues, are autotroph photosynthesis. If maybe the bacteria, for example, rhodospirillum, so that is photoautotrophs, you know. So then automatically the energy source is organic carbon, the energy source which is absorbed from photosynthesis. Okay, and if they are belongs to heterotrophs, they are absorbed from plant residues. Whenever the dry leaves, they are going to wet and decompose in the soil. that the plant residue can utilize the organic
వచ్చి అంటే వచ్చి
energy
అట్లనే
source
ఓడుడు
of
కదా
this
తర్వాత
organic carbon is plant
వస్తాను
residue.
కొంచెం
Okay,
తర్వాత
so then
నాన్నది ఉంది
in
మళ్ళీ
this process, the estimation of the fixation rate of the
18th కి అవుతుంది సిక్స్టీన్త్
nitrogen
పెళ్లి కదా
kg for hectare, kg for
15th
hectare in
కదా.
year. See here, so
18th
symbiosis
అవుతుంది
manner
నాన్నది
50 to 400 kg. Then association with their non-leguminous association, they are 100
Yeah,
or 10 to 200 kg per
వస్తాను.
hectare. And here, whether they are free living bacteria
అయిపోయాక
or
వస్తాను.
they are heterotrophs, 1 to 2 kg per hectare per annum. At the same time, whether they are autotrophs, maybe the photosynthesis bacteria, they are fixation of the nitrogen from atmosphere 10 to how much kg? Come on, 10 to 80 kg. 10 to 80 kg. 10 to 80 kg of 10 to 80 kg of nitrogen fixed by these three living bacteria. So symbiosis bacteria can ability to fixation of the nitrogen from atmosphere high amount of nitrogen. Are you agree or not? See here, 50 to 400 kg. But they are associated with non-leguminous plants, roots. They are fixation of the nitrogen from atmosphere 10 to 200 kg per hectare. But whereas in free living bacteria, very little amount of nitrogen is fixed, 1 to 2 kg and 10 to 80 kg per hectare. Am I right? No,
Yes.
you are not. Am I right? Is it right or not?
Yes, sir.
పిల్లలు వచ్చిండ్రు కదా.
Yes, sir.
ఏ పిన్ని రాదు.
This image is shows about
సాయి వాళ్ళు
the, how
వచ్చిండ్రు.
different
విష్ణు అట్లా
bacteria
వచ్చిండ్రు.
they are
వేరే
involved in this
సింధు వచ్చింది.
nitrogen fixation process. And my dear friends, remember here, this biological nitrogen fixation is also we can call as diazotrophy. What we can say that? Come on, tell me anybody. Diazotrophy. The method of nitrogen fixation we can call as diazotrophy. Which method? The biological being like microorganisms, they are involved in the nitrogen fixation process. The entire process we can call as diazotrophy. Diazotrophy. Di means two. Di means two. Azo means nitrogen. Trophy means living organisms. So the living organisms like bacteria, cyanobacteria, or some of the algal members, they are involved in the fixation of the nitrogen from atmosphere. That entire process we can call as diazotrophy. This diazotrophy has first clearly explained by Gastafson. Who discovered? Gastafson.
Okay, but Korea.
Which scientist?
But Korea.
My dear
Korea,
friends.
Korea.
Which scientist?
Korea.
Gastafson. Okay. Some here, see here. So some of the bacteria, maybe they are heterotrophs, maybe they are photoautotrophs. If they are heterotrophs, maybe they are free living bacteria, symbiotic bacteria. Okay. If they are photoautotrophs, maybe the free living bacteria, symbiotic bacteria. If they are symbiotic bacteria, they are associated with legume plants. They are associated with non-legume plants. If they are photoautotrophs, even though they are symbiotic, they are non-nodule forming bacteria and nodular forming bacteria. Okay, so these are this table or this flow chart is clearly explained how diazotrophy involved in the nitrogen fixation. Maybe the formation of fruit nodules. Maybe they are free living associated with the other plant parts. Maybe the roots or other internal parts of the plants. Okay. So here, the nitrogen fixation requires energy input. See here, my dear friends. So in this process, reduction reaction, so electrons must be added. Just because, why? Because sensitive to oxygen. My dear friends, remember, this nitrogenase enzyme very, very important. Nitrogenase enzyme is a very important enzyme which is also very sensitive enzyme, sensitive to oxygen. Okay. And it requires 13 kilojoules of energy per mole of nitrogen fixed. It is theoretically the actual cost of what we can say that 15 to 30 grams per gram of ammonia produced. Okay, then the assimilation, the final assimilation of ammonia into organic forms takes 3.1 to 3.6 gram per the total nitrogen fixation. See here. Then in this process, nitrogenase enzyme is involved. Am I right? Nitrogenase enzyme. Okay. So in prokaryotic living organism like bacteria and cyanobacteria, so rhizobium fixed nitrogen in symbiotic association with leguminous plants. Are you ever heard of leguminous plants, my dear friends? Give any examples, one or two examples, very quickly. Very quickly. Remind immediately. Okay, very good. So all pulses, they are belongs to leguminous plants. You know, Bengal gram, green gram, groundnut. Okay, soya bean. So they are belongs to leguminous plants. So these leguminous plants associated with rhizobium leguminosarum bacteria. Okay, so these rhizobia fix nitrogen for the plant, and plant provide rhizobia with carbon substrates. Okay, carbon substrates. Then all nitrogen fixing system appear to be identical. So all are identical. Here they require nitrogenase enzyme. They are along with nitrogenase enzyme. My dear friends, remember here the reductant also involved. Reductant means reduced ferridoxin. I'm already told that this nitrogenase enzyme is involved in the nitrogen fixation process. This nitrogenase enzyme is contained two specific components, that is the iron molybdenum, that is a ferrous molybdenum or iron molybdenum segments are present in nitrogenase enzyme along with this ATP. And even though oxygen-free conditions are most important. So if oxygen is present, the nitrogenase enzyme is inactive position. Without oxygen, nitrogenase enzyme is very, very active. If oxygen is present, then this nitrogenase enzyme is very sensitive to oxygen. So that is malfunction or not function properly. So oxygen-free conditions in environment is mandatory in this of NIF genes. NIF genes means nitrogen fixation genes. What is NIF genes, my dear friends? Nitrogen fixation genes. ఇది కనబడుతుందా మీకు? ప్రొకారియోట్స్ లో జరిగే మెకానిజం గురించి చెప్తున్నాను. కనబడుతుందా మీకు ఇది?
Yes, sir.
Outside.
Okay, it is visible. The slide is visible.
Yes, sir.
Okay, so the enzyme, so oxygen sensitive enzyme, that is nitrogenase enzyme. Very, very important. Nitrogenase enzyme very sensitive to oxygen. If oxygen is present, nitrogen is in inactive position. If oxygen is not present or anaerobic condition is there, then automatically nitrogenase is in active position. Okay, active position. But whereas, see here. But in aerobic condition means oxygen presence. So if oxygen is present, nitrogen is not properly working or inactive position. Remember, my dear friends. Okay. At the same time, the reducing power is produced by NADPH, that is the ferridoxin, via an iron molybdenum center. Iron molybdenum center. MO means molybdenum. Fe means iron. Okay. same time, plant genomes lack any genes coding this enzyme, which occurs only in prokaryotes like bacteria, cyanobacteria. If even within a bacteria, only certain free-living bacteria like Klebsiella, Azospirillum, Azotobacter, they are in free-living bacteria. But some of the bacteria they are associated with legume plants like Rhizobium leguminosum is there. But here, what we discussed, Klebsiella, Azospirillum, and Azotobacter, these are all free-living bacteria. Along with this, some of the blue-green algae like Anabina, Oscillatoria, okay, nostoc, and a few symbiotic rhizobial species are known nitrogen fixers. So these are all nitrogen fixers. Nitrogen fixers means the living organisms, the microbes, which they can ability to fix the nitrogen from atmosphere. Are you agree or not? Are you follow or not, my dear friends? Respond quickly. స్పీడ్ అవుతుందా స్లో చెప్తున్నాను స్పీడ్ అవుతుందా స్లో చెప్తున్నానా
నేను రీసెర్చ్ చేయించలేదు.
అదే ఎట్లా చేసిన
nobody can respond.
మంచి ప్లాన్ పెద్ద పెద్ద
ఓకేనా
200 పెట్టాను
వింటున్నారా
Okay, sir.
ఓకేనా బయట పోతే
వింటున్నారా
గూగుల్ పే కాకపోతే ఒక నిమిషం పనికొస్తే 200
one side కాదు two side మన involvement ఉండాలి my dear friends. Remember the another nitrogen fixing association exists between an actinomycetes, that is the Frankia species. Actinomycetes species is a special type of bacteria. Actinomycetes is a special type of bacteria which are very related to or very nearer to very closer to fungal members. So that bacteria we can call actinomycetes bacteria. The actinomycetes bacteria example is Frankia species and also Alnus species. So these species are non-leguminous plants which they are produce the root nodules in their roots.
ఎక్కడిది
Okay. So Frankia and Alnus. And the another important slide. This slide is visible. The enzyme nitrogenase catalyzes the conversion of atmospheric gas into ammonia. See here. So this is nitrogen and this is oxygen. So nitrogen and oxygen both they are associated to produce 2 moles of ammonia. Here, this is visible 2 moles of ammonia is visible.
నేను అంటే చూస్తా అయ్యో
Okay. So the FEP reaction seen simply enough in the atmospheric nitrogen, how much percentage of nitrogen is present in atmosphere? How many? 78% of the nitrogen is present in the atmosphere. Highest amount of the gaseous substance, highest amount of the elements which are present in atmosphere, that is nitrogen. Okay. Then some of the friends is asking me personally, sir, why this enzyme is so important? This enzyme means nitrogenase enzyme. This enzyme means nitrogenase enzyme. So the strong means the triple bond of nitrogen makes this reaction very difficult to carry out efficiently. So the efficiently the nitrogen or molecular state of the nitrogen between these two nitrogen atoms, the triple nitrogen hydrogen bonds are there. So the nitrogenase engine contains 60 moles of the ATP. ATP means energy, energy molecules. So these energy is utilized and break down or
ఫోన్ తీసుకొని ఒకసారి
or nitrogen
ఏ కన్నయ్య
ammonia. So
ఫోన్
which
ఇంట్లోనే
makes
రావు
one
ఇది వచ్చి
of the
ఫ్లాష్
most
లైట్
energy expensive.
దీనికి ముందుకొచ్చి రావట్లేనా చూద్దాం పెట్టు
And what is nitrogenase complex? We know that nitrogenase is the complex enzyme, so which contains either molybdenum or ferrous molybdenum substances. You know,
ఏ కదలకురా
and my dear friends, remember here nitrogenase
ఏ
complex is two
అదే రోజు
components.
కూర్చుంటారు అంటాడు
That is the nitrogenase
దగ్గరికి
and nitrogenase. See here, so nitrogenase reductase is a 60 kiloton
ఇంటికైతే
homodimer with a single
నవ్వుతాడు కదా
ferrous sulfur cluster. My dear friends, remember, so it is the nitrogenase reductation
కొట్టినట్టుంది
with a single iron and sulfur cluster is present. Whereas And remember here, my dear friends, nitrogenase is an enzyme which
అయ్యో
is
వీడు
very
హోంవర్కే
sensitive
రాయలేదు.
to oxygen molecule. And the nitrogenase enzyme binds with magnesium ion along with ATP, adenosine triphosphate. And 4 ATP is required per pair of electrons transfer. How many ATPs? 4 ATPs. Then 4 ATP means in our equation how many ATPs are required? 16 ATPs are required. So then how many pairs of electron transferred? 4 pairs of electron transfer. Am I right or not? So in this process, the reduction of nitrogen to ammonia,
అమ్మ బర్త్డేకి కేక్
yeah,
ఎందుకు కట్
protons or
చేస్తారమ్మా?
hydrogen requires 4 pairs of
వేరే
electrons.
cut చేస్తే
So
పాడైపోయింది.
16
ఏ
molecules
తప్పు
of ATPs are
కేక్
consumed
బదులు
for
వేలు కట్ చేస్తే
1
అయిపోతుంది.
mole of nitrogen or dinitrogen. See here, so this is a equation, chemical equation. You know, 16 electrons,
Yeah,
and 16 protons.
చాచి
So this biochemical energy required here. The triple bond present in between two nitrogen atoms. How many bonds are present?
తీసినము నొప్పి తగ్గుతుంది.
Triple bonds.
తీయకుండా కూర్చో.
See,
మరి
these
finish
triple
కావాలి.
bonds is going to cut down
తీసుకోదు.
or break
మెల్లగా
down by
అదన్నే తీస్తుంది
nitrogen
కదా.
engine by the uses of the how 16 ATPs of amount of energy. Then it is converted into the end product or the final product, that is 2 mole of ammonia. 2 mole of, so 1 mole of dinitrogen is going to convert it into 2 mole of ammonia. So in this process, 16 ATP energy. How much ATP? 16 ATP energy is required. So it is a reduction process, my dear friends. Reduction process. So the nitrogen reduction to ammonia
వీడు ఏం
is
చేస్తుంది?
thermodynamically favorable. Thermodynamically favorable. And here, however, the activation there of breaking the triple bond between the two nitrogen atoms is enormous.
అమ్మ శ్రేయ మీనాక్షి
Utilize
చేయకు.
16 ATP amount of energy. Okay. Then in this nitrogenase
మీనాక్షి
agent,
తా
220 kilodalton, you know,
కట్టు హ్యాపీ
and
నేను అక్క
in previous
ఆహా నాకు నాకంటే
so
నువ్వు
the
క్లాస్ పెద్ద కదా
nitrogenase
ఏమవుతుంది
complex,
చెప్పు
is an enzyme,
ఏమైతే
is a
చెప్పు?
protein,
ఏం కాదు
so which contains 16 kilodalton
నేను
homodimer.
చేసినప్పుడు రా అంట
With a single
అనొచ్చు
iron
2 క్లాసెస్ గా నాకన్నా
cluster.
చేసి
But
4th
whereas
త్రీ అంటాడు అనొచ్చు కదా నేనంట
the nitrogenase
త్రీ
agent
వన్
is a 220
మీనాక్షి
kilodalton heterotetramer.
అన్నది.
That is homodimer, nitrogenase
KR
reactive
and
enzyme.
KR
నగరమా?
Homodimer.
కేన్నారంటే కరీంనగర్.
Okay, but here the
అమ్మ మీనాక్షి వాళ్ళు
nitrogenase agent 220 kilodalton at the same time
Kandera.
heterotetramer.
అందుకే కండేరా
Heterotetramer. Here
ఏమిది
the
పిన్నిస్తే అంటే?
nitrogenase
కండేరా
agent
అంటే ఏంటిది
contains
ఇంటి పేరు ప్రియరబాటు
4 moles of iron, sulfur
ఆ కొత్త
substance only. But whereas nitrogenase agent contains 2 molybdenum
మంచిగా పడక
and
పెట్టి
32 ferrous and 30 equivalents of acid labile sulfide. That is FES cluster.
కట్ చేసి
These are present in
నేనే
reductation.
వేసిన నీకు నచ్చదు ఏం
And
నచ్చదు
at the same
అది
time,
టక్కన
sorry,
ఎక్కుతుంది
nitrogenase
పనులు
agent.
ఉంది కనుక
At the same time,
ఎక్కలేదు
4 iron
అదంతే
and 4 sulfur clusters plus 2 ferrous molybdenum cobalt
గుండె ఇప్పుడు మనకి
and
కొమ్ములు వస్తాయి
iron molybdenum cofactor is present in this nitrogenase agent. So nitrogenase is a slow 12 electron pairs per second. At the same time, only 3 molecules of nitrogen per second.
50-40
So
అక్కడ
this,
ఒక
this is
బాక్స్
the 3D cluster. We
తమ్ముని
need not to
బుక్ లో రాయి వాడు
tell
డౌన్లోడ్
about
చేస్తారు
the genes
ఏం
and
చేయాలి
products.
నువ్వే
See here, this is how
ఇలా
iron, sulfur and molybdenum electron transfer cofactor is binds here. Okay.
నాకు కూడా
How many types of the nitrogen
Manchi
fixers
gundi,
important in
manchi
forest
gundi.
soils?
నేను చెప్పు
Forest soils.
birthday
How many
lala.
nitrogen fixers
Na
see here?
book.
These are
Na
3.
birthday
That is the
Pani
cyanobacteria,
nuvu.
heterotrophic
నా
bacteria,
బర్త్డే
and symbiotic bacteria. So cyanobacteria
మరి అయితే నీ
maybe
బర్త్డేకి
the nostrils, maybe
మరి
the
ఎప్పుడు
anamina,
కావాలి
maybe the oscillatoria species,
అన్నయ్య
they
బర్త్డేకి
are
కావాలా
associated with
కొత్త సైకిల్ కొన్నదా
other living organs, other
అట్లయితే
real
నాకు
plants,
డింగచిక
or
ఒకళ్ళు
maybe they are
కరీంనగర్ సైకిల్ తప్పితే
forms.
కుక్కలకు
Whereas
భయపడకుండా పోతా
living bacteria, they are
డాగులు
even though
భయపడకుండా
living
పోతా
bacteria
సైకిల్
are
నుంచి
associated with
ఇంత స్పీడ్
rhizospheres.
కావాలంటే అంత
Rhizospheres
స్పీడ్
means
ఓకే అంటావా
area or part of the roots.
ఏమో
That use energy from decomposing
కూర్చో.
organic matter to fix nitrogen. Protect nitrogen by rapidly converting oxygen to
అమ్మ
carbon
నాకైతే
dioxide
రింగ్
through respiration.
ఏం
Then
రాదు
the
దీంతో
third
ఒట్టి తొక్కడం
types of the
తప్ప
nitrogen fixes, they are symbiotic bacteria
చేతుల్లో
like
తొక్కడం
rhizobium, bradyrhizobium.
సైకిల్
Okay, for legumes and frankia, for non-legumes, even though the nodules contains a form of hemoglobin which binds oxygen and protects nitrogen. So, what we discussed in previous slide, nitrogen agent is oxygen sensitive. So, hemoglobin, that is the red
సిస్టర్
hemoglobin,
పని
is
చేసి
acted as an oxygen scavenger. Remember, my dear friends, oxygen scavenger is hemoglobin.
నేనేం పెడతావు
మీనాక్షి
So, liquid
Happy
plant
birthday,
is
Meenakshi.
release that red colored,
హ్యాపీ బర్త్డే
the
మీనాక్షి
red
అక్క
pigmentous colored,
చెల్లె
area we can call as leg hemoglobin,
Happy birthday,
which is red
Chella.
color, which binds oxygen and acted as a oxygen scavenger. So,
హ్యాపీ బర్త్డే మీనాక్షి
mechanism
Ta-ta.
leads to the very active way involving conversion of the atmospheric nitrogen to usable form of the ammonia or nitrates of this nitrogen.
హ్యాపీ బర్త్డే
See here.
మీనాక్షి
So the classification clepsydra.
ఓకే
So this system is there now.
ఆగు ఆగు
Started on when no fixed nitrogen.
చూడవా
It is
ఏది
mandatorily anaerobic condition, and even though the temperature below 30 degrees Celsius, temperature below 30 degrees Celsius, and even though the
హ్యాపీ
nitrogen
బర్త్డే
converts nitrogen to ammonia.
హ్యాపీ బర్త్డే
See here, so this matter we can observe in this
మళ్ళీ పెట్టాలి
image. The slide shows you this is the nitrogen is injected.
చెప్తాను
Uses of this nitrogen is injected.
తాకుతే ఏది పట్టి మెంటలోడా ఏది పడితే ఇంత కొడతారు ఎంత బరువు ఉంది
Is
నాన్నకి చెప్పిపోయి ఇంత కొట్టి
this
తల
is
మీద
the dinitrogenated.
మస్తు అలుగుడు కూడా
Nitrogenase
వచ్చింది
and this is cell metabolism. And then after the mechanism, whenever the process is continuously progressive manner, then nitrosylation of the dinitrogenase reductase is actively involved in this process. Okay, so in this process, see here, so nitrogen, dinitrogen is converted into ammonia. It is a usable form. So this is the dinitrogenase agent. At the same time, so
చూడు
the
చూడు
mechanism, dinitrogenase reductase mechanism we are observed is like.
ఏం కాదు పోతుంది బరువు ఉంది అంతే అది
So
దీన్ని కొడతా
this is the
దీని తల
legume
మీదకి
plants. Have you ever seen
Charging
this legume
network.
plants?
ఇది కొడతా
Hey,
Root
charging
nodules. We are
network.
observed in this root nodules, you
నేను
know.
చెప్పేది అయితే ఇదే
Root
చెప్పు
contains nodules.
అది నువ్వే ఇష్టం ఎందుకు తీయాలి
legume
మాకు అసలు
plants,
ఏది ఉంటుంది కొడతాడు
they
చార్జింగ్
are-
పెట్టు
They
లేదు
are having the root nodules or produce the root nodules
అవి
by
గట్టి
the
నిమిషం డాష్
involvement of the
ఎందుకు
rhizobium
నిమిషం
bacteria
నాది
or microbial organisms. So the exchange of the nutrients during rhizobium legume symbiosis. See here, this is the symbiotic metabolic how to utilize both the association, the symbiotic association, how to involve to exchange the nutrients for survivalence process. So in this symbiotic nitrogen fixation process, this is the rhizobium legume association. This is one of the Fabaceae member or leguminaceae plant roots which we are observed in this image, right side. So these are the root nodules which is produced by bacteria. Okay, so bacteria associated with certain plants families, mainly, primarily, especially legume plants. And the species make the largest single contribution to biological nitrogen fixation in the biosphere. See here, it is a pea plant can ability to sustain or produce number of root nodules with the help of rhizobium leguminous organ. Okay, so in this inside the rhizobium, inside the root nodule, when we are cut down or break down or take a section, the root nodule inside we are clearly observed. Pink, light color pink or pink color, light pink color, we are observed. That is nothing but leg hemoglobin, a protein that carries oxygen to the bacteroids. Oxygen to the bacteroids. So that leg hemoglobin is acted as an oxygen scavenger. Oxygen scavenger. See here, it is a soybean plant. So soybean plant contains number of, we are observed number of
పిచ్చి వేషాలు వేస్తే కన్నయ్య
on the surface of the roots. Okay. So
నీ తీసుకోడు వేయలేగా
nodules develop by
అట్లా నీ కొట్టిస్తా ఆగు
broad rhizobium.
ఏ ఇస్తావు నువ్వు పిచ్చి వేషాలు వేయకురా
One
కట్ట
bacteria,
తీస్తున్నా
one bacteroids are the
ఎందుకు
what we can
పడేయాలి
say that root nodule pods we are
అది
observed in
తీసే
the
కష్టం అయితే అన్ని పడేసి
So
పెడతాను
one
మంచిగా
root nodule we are observed. See here.
ఏ నాకు తీసిన కష్టం మీరు మంచిగా పడేసుకుంటారు తీసిన కష్టం దానికి కట్టేసుకుంటుంది వద్దు ప్లీజ్
And this is the nodule
కన్నయ్య
apex
చెప్తే
zone.
వింటాడు ఐ నో దట్
And this is O2 gradient zone.
కన్నయ్య
This is
చెప్తే
the O2
వింటాడు
gradient is
నీ
there.
బుక్ పోరా
But inside is less.
చెప్తే చేజాడి పేజ్ ఇప్పుడు మీరు
నేను రాస్తా.
రాసుకొని
Just
నువ్వు
because of the leg hemoglobin
చేజాడి పేజ్
absorbs the
ఉంది హాస్టల్లో వేస్తే ఉంటది
even though present
అది
of
ఇంకేదని
inside the
ఏమంటారు దాన్ని
nodule.
ఏమంటారు
Okay.
కో ఏదో అంటారు
Then see here.
పైన
This is the
ఎక్కించిన
leg hemoglobin.
నాన్న
See here,
ఇంకో పైన
the
ఎక్కేయడానికి
కోదండం.
nodule. See here.
మంచి
So
గుర్తు
this is the
కోదండం ఎక్కించడం
external
ఉంటుంది.
aperture or
చేజాడి
external part of the
చూద్దాం
root.
చూస్తాను
Nodule.
చేయరు
So this is
స్కూల్లో
the
వాళ్ళు
cortex
చేస్తారు.
of the
చూద్దాం.
root.
చూద్దాం
At the same time, this is the
పైన ఇట్లా ఉంచుతారు
light
క్లాస్ కూడా
blue
పైన
color,
అందరు మిమ్మల్ని
blue color
చూసుకుంటున్నారు
region.
వచ్చి
They are
బ్యాడ్
to
బాయ్స్ అని
vascular
చూద్దాం.
అట్లనే వినాలి క్లాసులు చేయి ఇట్లా కట్టేస్తారు పైకి
contains
రాయమన్నారు
complex tissue.
వినమంటారు అడుగుతారు క్వశ్చన్
These are
అడుగుతారు
present
Up.
vascular tissue.
కొంచెం అయినా ఉంటాను చూద్దాం చూద్దాం.
Black color, that is the
నేను అన్ని
included
మాటలే
tissue.
జారి పోయింటారు మీరు
So whereas
కూడా అంటారు టీచర్ని.
ఇది వద్దు అది చెప్తే
ప్రిన్సిపల్ టీచర్ కి పోయి చెప్తా నేను
they
కూడా
are observed.
ఏమని
The
గొడవ
plant
చేసి
cell
కలతం
division
కొడుతుంది.
is a very
నేను స్కూల్
rapidly
మానేస్తాను
plant cell is
చెప్తా.
going to
ఎవరు?
division.
మా టీచర్
Division
కొడతాను నేను
condition
చెప్తాను.
and newly
ఇవ్వ- ఇవ్వండి.
condition with low nitrogen fixing abilities. Here, the
భయమే లేదు.
light
అదే
brown or
చేయాలి
pinkish
కదా.
color region we are
నేను
observed.
చెప్తే
Then
వేయకుండా
mature
వినేసారా
zone
మరీ
is
ఘోరం అయిపోయింది
directly pink
బుక్
color zone
తెచ్చుకొని
we are
మీరు
observed.
ఏదైనా
High
అదే ఎట్లనే
nitrogen
చేస్తే మొన్న
fixation
వేసినప్పటి
ability which we are
అప్పుడు
observed
ఒక
in
రోజు
this
వేయాలి. తినంటే
zone. That is the
తినకండి.
mature zone. Where at the
వేసిన
same
ఒక్క
time the
వేసినా?
senescent zone is also there. So
ఒక రోజు
this
వేసిన
senescent
బ్యాడ్
zone,
పెట్టి
the
క్రికెట్
light
ఆడినాం
pink
కదా
is,
ఒక రోజు.
pink color is going to dark red or red color zone we are observed. That is the low zero nitrogen fixation ability we are observed. So this
అమ్మ ఆ రోజు
bacteria is,
నువ్వు నన్ను బ్యాగ్
hydrophobic
తీసుకొచ్చినావు
bacteria,
కదా
even though they
సైకిల్
are
రోజు
sorry,
అది
they
ప్లాన్
are,
మొత్తం
they are autotrophic
కెమికల్
bacteria by because they are photosynthetically
ప్లాన్ వేసి
active.
మరీ
They are prepared their own food material in presence of the sunlight. So these photosynthetic cells are
వాళ్ళు
here.
ఎట్లా ఎట్లా
So
ఉంటుండ్రో
these,
ఆటోమేటిక్లీ
these nostoc or anabina
Yeah,
cells,
yeah,
they
sorry.
have, even though the
ఇది
heterocyst
మనదా?
also present. So
ఏం కాదు
the
టచ్ స్క్రీన్
heterocyst
అయితే ఏం
cells
కాదు కదా.
is the specialized cells which get ability to fix the nitrogen from atmosphere.
అది
That
వాళ్ళు
the heterocyst is
వేరే
one of
ఇది
the
మనం
important
చేస్తున్నారు.
cells of the trichome of
వాళ్ళు
the
ఇక్కడ
green
ఎట్లుండ్రు?
algal members.
వాళ్ళే చేస్తున్నారు వాళ్ళే
this is
క్లాస్
how
చెప్తున్నారు
nodule,
ఇది
how
ఇక్కడ ఎట్లుండ్రు?
root nodule is
నేనే
produced.
ఓపెన్ చేసిన కదా
So the nodule
నేను
formation,
అందరినీ జాయిన్
how
చేసేది
to
నేనే
formation of the root
అట్లా
nodule.
కాదమ్మా,
అట్లా చేస్తున్నారు.
బీచ్లు
Nodulation
ఎట్లుండ్రు
process. So in
వాళ్ళు?
this nodulation
తీయరు.
process,
వెనకాల
so these are the
వేస్తున్నారు
steps
అంతే.
we are observed. The first one is the,
అట్లా పెయింటింగ్
are you follow my dear friends?
అది వెనకాల
Are you
పెట్టుకోవచ్చు
feel
సెట్టింగ్స్
good?
బ్యాక్ సైడ్
Are you
అది
follow?
నాకు కూడా వచ్చి నాని ఫోన్ లో
Okay. So the nodulation process,
అమ్మమ్మ
the chemical
చెట్ల
interface
గురించి
of the root
ఏమేమి
and rhizobium,
చెట్టు
and the
గాలి
second step, that is the root hair
పచ్చగా
curl,
చెట్ల
and
పండు వస్తాయి
that root hair curl also we can call it shepherd crux,
సాల్ట
and formation of
సాయిల్
infection threads,
సాయిల్ టాకర్
and fourth one is the invasion of the roots by rhizobia, and fifth one is the nodule tissue forms, and sixth one is the bacteria convert to bacteroids and begin to form nitrogenase engine, and lastly the seventh one legume provides rhizobia carbon, rhizobia, my dear friends, remember legume provides
బన్నీ
బనియన్
rhizobia with carbon.
కిన్నే
At the same time, rhizobium provides the legume which gives you nitrogen. So nitrogen is provided by rhizobium, and carbon is provided by legume plants. Are you follow or not?
ఎస్ సార్.
అమ్మా వినబడుతుందా?
ఎస్ సార్.
లిగ్యూమ్ మొక్క అనేటువంటి కార్బన్ అందిస్తుంది. కార్బన్ కంటెంట్ ను బ్యాక్టీరియాకి అందించేది లెగ్యూమ్ ప్లాంట్. బ్యాక్టీరియా ఏం చేస్తుంది? లెగ్యూమ్ ప్లాంట్ కు నైట్రోజన్ అందిస్తుంది. So this association organism get benefited. So this type of association we can call as symbiotic association. Okay, so this chemical recognition of the roots and rhizobia, root hair curling, formation of the infection traits, expansion of root by rhizobia. So these are the 7 steps we are observed. Okay, so this is today's class, my dear friends.
ఏది నాకు ఏం 19 అంటే 7 అయిపోయిందా?
Okay, so these are the how root nodules are produced on the surface of the root.
అన్నందుకు అమ్మ రోజు తొందరగా లేట్ వస్తావు కదా.
9 మీతో
My dear friends, I will stop here. If you have any doubt, please ask me, my dear friends.
మై డే ఫ్రెండ్స్ అతను అడుగు ఏమన్నా డౌట్స్ ఉంటాయి అడుగు.
No doubt.
నో సార్, థ్యాంక్ యూ.
ఆ డౌట్స్
So
ఉన్నాయి.
here at the same time, some of the legumes they are belongs to other than bacterial members or other than legume plants also involved. See here, alfalfa not belongs to legumes, you know. So clover, soybean, beans, peas, sesbania. So these are all host plants. So bacterial symbiote is different. Rhizobium meritoli, Rhizobium trifoli, and then Bradyrhizobium japonicum, Rhizobium caesioli. So Rhizobium leguminosaurum, Azorhizobium, Colonodons. So these are the other bacterial symbiotes which they are involved in different plants. They are belongs to Leguminaceae family members. Okay, right? So I will stop here, my dear friends. Tomorrow we will going to discuss about the other new topic. I hope you clearly understood
మీ
today's
సార్
topic.
పేరు ఏంటి అమ్మ? తిరుపతిరావు సైనింగ్ ఆఫ్. బాయ్ బాయ్.
Even a doubt to study.
Ten by nine,
Any doubts?
నో డౌట్ సార్.
No doubts? Okay.
నువ్వు కొద్దిగా
Tomorrow we will meet. Okay, my dear friends, I will stop here. Madam ji, Manjula Madam ji.
ఎస్ సార్.
Okay, thank you very much.
कभी याद करे तो तुम्हारा.
Madam,
ఏ మాట్లాడకు. ఓకే సార్, థ్యాంక్ యూ సార్.
right. Okay, with your permission, I leave today class.
ఎస్ సార్, ఎస్ సార్.